There's trainees in the room, and he mentioned my most recent work, but I originally started as a surfactant biologist, which is how I understood type 2 biology. And so I'll start with a quote from Yates. And for the trainees, your career is going to be like a spiral staircase where you always come back to the same point. So here, but you're hopefully going up to a higher and higher level. And so for all of us here, we're going to have this repetitious journey. So I'll take you through a little bit of this journey because the talk I gave yesterday at the Pneuma Trieste was more clinically related to therapies. I wanted to delve back into the cell biology, which is sort of my former life, and then tap into some of the expertise here as well. So from there we go. So I will start with the disease because I am a physician scientist, and this is the disease that I see and take care of and supervise the research program for. It's idiopathic pulmonary fibrosis. It's one of a number of fibrosing interstitial lung diseases. The pathology is down here. You can see that these are fibroblastic foci. The interesting thing about this is that there are areas of fibrosis next to areas of normal lungs. So there's temporal and spatial heterogeneity as the disease progresses. The diagnosis now is made mostly with a CAT scan. So you can look at this and say, this is a UIP-like pattern correlating with this, and this is a diagnosis. This is some of the epidemiology down here. And for us as physicians, it's very frustrating. So this is back to the original marketing for the two drugs that we have. These are antifibrotics. On the left is pirfenidone, and on the right is nintedanib. They're both pleiotropic drugs. Pirfenidone has some TGF-beta ameliorating properties, and this is a triple kinase inhibitor, somewhat of a dirty kinase. And you can see that when you compare the decline in lung function here, it's actually, we only slow the progression. So this looks like the drugs we have in 2023 are like what we had in 1960s for cancer, single drugs that basically slowed the progression but did not cure cancer. And so major problem. And I think partly it's because we didn't understand the disease that much. And so from the surfactant biology that I did, and through some of the things I'm going to tell you, what we now recognize, and this is a concept that's throughout the field, it's not my concept, but IPF is a polycellular disease. So it's not a disease just of fibroblast expansion. We now know that there is intrinsic and extrinsic damage to the epithelium. We're going to talk a lot about this. There's also a phase of this where there's amplification, specifically the recruitment of things like the LY6C monocytes, which are profibrotic and increase this mesenchymal crosstalk and epithelial crosstalk. So very much a polycellular disease. And so if you look at the drugs that we have now, the two I just showed you are antifibrotics. They act down here. The drugs that are coming up in phase 3 clinical trials, so PDE4 inhibitor and LPA antagonist, are thought to also work downstream, although I think we have some data now that maybe some of these will actually help with upstream. And some of these other ones, of course, we know TGF-beta is important for myofibroblast expansion, and CTGF is important for epithelial now, mesenchymal crosstalk. Again, we don't have any drugs specifically targeting either regeneration or improvement in lung epithelial function here in terms of turning off the driving forces. So I studied the type 2 cell for about 35 years. This was a phrase that was coined originally by Mary Williams and Bob Mason, that this is the defender of the alveolus for so many reasons. It's important for barrier function, and that's shown here. We have apposition of type 2 cells and type 1 cells shown in differential staining. You have the appearance here of host defense, which I won't talk about today. Highly metabolic cells, we won't spend a lot of time on this, but they have a huge amount of mitochondria, and that's because they have to make, package, and synthesize surfactant, which is incredibly important for keeping alveoli open at low lung volumes. And so premature babies have defects in the maturity of the type 2 cell and the surfactant system, and so that's what you need to keep the lung open. They make surfactant proteins. They package lipid into lamella bodies. This is now a red staining shown here. And so, we're going to talk a little bit about this. So this is a cell at risk. They're also stem cells. So we were talking a little bit earlier about the Christine Markowski study with Bridget Hogan, but basically if you take type 2 cells out, lineage tag them, put them into culture, follow them in organoid culture, they turn into type 1 cells. So they can self-renew and differentiate. So they meet all the criteria of a progenitor cell. If you actually ablate these by a variety of mechanisms, whether you choose to do this with diphtheria toxin or with a bystander effect from ganciclovir metabolism, as you deplete these beyond 50% of your type 2 cell burden, you actually then can initiate spontaneous fibrosis. This is a study from Michigan, Ralph Sisson, and this was Barb Driscoll's work out at UCLA. And then we also know that senescence occurs, and this is actually part of a study from Barry Stripp, where they looked at all of the cells coming out of IPF lungs and found a huge amount of scoring in many, many different cell populations for the presence of cellular senescence. So their absence leads to fibrosis, their senescence probably is just as bad. And so these are stem cells which need to be protected. So we have to go back now and talk a little bit about surfactant biology to understand what I'm going to tell you next. And that is surfactant that is secreted by the type 2 cell is mostly a phospholipid mixture. About 90% of it is a variety of phospholipids, mostly saturated and unsaturated phosphatidylcholine. But it needs, in order to work biophysically, and that is to lower surface tension at an air-liquid interface in the distal lung, it actually needs either detergents, or in this case, we have natural detergents in the form of two very hydrophobic proteins, surfactant proteins B and C, which actually intercalate into the lipid monolayer. It can actually facilitate the packing of the polar head groups and facilitate some of the spreading of this. And so without these, SPB deficient humans die of respiratory failure very early in life or need lung transplant. I point this out because we're going to talk about SPC. It's a 35 amino acid peptide. If you look at the structure of this is all valine, isoleucine, hydrophobic amino acids mostly, and that's so that it can form an alpha helix in a lipid environment. So this creates a nightmare for a cell in terms of biosynthesis, in terms of its intercalation in the membranes, its trafficking, and its targeting. In fact, when left alone in the alveolar space, that is in cases with patients with pulmonary alveolar proteinosis, which is a defect in the recycling of surfactant, it actually forms amyloid-like properties, and that's shown here by its insolubility to SDS and its ability to stain for amyloid, shown here. So this has to be dealt with, and the type 2 cell is a fairly, as I said, a complicated cell. So what nature did is it packaged this protein as a protein, which has a short amino terminus. It's a type 2 protein, so when translated, it actually has a short amino terminus in the cytosol, and then the mature protein that eventually gets secreted is actually intercalated into the ER membrane, and then there's a large folding domain down here. Its function is not well understood, but actually improves the trafficking, and again, longer carboxy tail. There's 4 regions. We've published before that this is a targeting domain, and you need this because after synthesis, this goes through a fairly complex biology of exit from the Golgi, packaging into the multivesicular body, and then the generation of this lamellated lysosomal related organelle, which is the lamellar body, which has a acidic environment fueled by a protein ATPase. There's 4 cleavages, which we published on extensively in terms of multiple enzymes. There's aspartic proteases, as well as cysteine proteases that process this protein. All of this has to take place distal to the Golgi. So if you take cells, and actually we did this in the old-fashioned way with 35S methionine labeling back in the day, but if you do pulse chase, you can see that with epitope-specific antibodies, you can actually get processing to the various intermediates. If you block this exit from the Golgi with Profeldin, you get no processing. So targeting equals processing, and that becomes important to understand some of the mutations that we're going to talk about. So we've also published extensively on trafficking for this. And as I said, we discovered that there's a trafficking motif right here. So we basically make GFP fusion proteins. We did this in A549 cells. I don't recommend them as the ultimate model for a type 2 cell, but this is what we had. Or in HEK cells, you make a GFP fusion. You can get transport of these into cytosolic vesicles, which label with lysotractors, so they'll traffic to lysosomal related organelles. If you do some of the protein bashing, we localized the targeting motif to this SPPDY motif in the amino terminus. This allows it to interact with an E3 ligase, which allows this to be monoubiquitinated at lysine 6, which then is a targeting motif for getting many proteins to lysosomal related organelles. And so if you delete this, you now get ER retention shown here, no trafficking. We pointed out these cysteine residues down here. Again, this will be important for the disease variants I'm going to show you in a second. And with these, if you actually mutate one of these or both of these, you end up with initially ER retention, but eventually the formation of intracellular aggregates. This is a membrane-free aggregate that is adjacent to the nucleus. brought there by the microtubular organizing complex. You can block this with the cortisol, and this is an agrosome. So this is yet another garbage pit, basically, for misfolded proteins. So now getting to the disease, and so as I mentioned, there's 4 domains. Over the years since 2000, there's been over 60 mutations in surfactant protein C that have been associated with interstitial lung disease. The majority of these are in the carboxy terminus, shown here. And many of these, the original one was a deletion of the entire exon 4, which took out cysteine 120, 121 in the human. This one was in a large kindred of patients from Vanderbilt, which was actually a non-conservative substitution at codon 188. Terminal L188Q, that cysteine 189 is responsible for this. And so, and this is what we're going to talk mostly about today. I'm going to spend a lot of time on protein quality control. We're going to use this as a substrate to understand this. These are interesting. We will probably, if we have time, just mention these at the end. These are actually trafficking mutants. Nobody knows why this is true, but these actually do not aggregate or stay ER retained. I'll show you. They actually end up being miscycled to the plasma membrane and affecting other quality control mechanisms like macroautophagy. And so, in order to understand how you would handle these mutants, we just need to talk a little bit about how type 2 cells handle misfolded proteins. What are their proteostasis repertoires? Well, they have all the classic ones you would think about. And that we thought originally they were reasonably well balanced, but using A549 cells and HEK cells and other things. But if you actually look at this deeply, we looked at this both in isolated mouse type 2 cells, as well as we had a program for a while for the isolation of human fetal type 2 cells and their maturation in culture. Both of them, this is chymotrypsin based activity for proteasome activity, compared to HEK 293 cells, which are incredibly robust in their proteasomal activity. They have a fair amount of this in terms of chymotrypsin activity, both the mouse and the and the human. They also have the ability to perform macroautophagy, so you can stimulate this with an mTOR inhibitor like Torin, and you can get turnover of the classic LC3B protein from LC31 to LC32 in both cell types. And if you inhibit this with proteasome inhibitors, the majority of the substrates in the type 2 cell really are degraded by the proteasome and not by the macroautophagy pathway. So vancomycin will inhibit acidification and macroautophagy. But if you look at the appearance of polyubiquinate proteins, really the most robust responses with MG132 or other proteasome inhibitors lack the cysteine. And so what this suggests is that the majority of substrates in a secretory cell like this, in a polarized epithelial cell, are really read in real time, much like in the pancreas. And so places like the brain rely a lot more on macroautophagy to get rid of protein aggregates, but most of this is read in real time. And so what's interesting, if that's the most important way to get rid of a protein, surfactant protein C has really figured out a way to short circuit the system. And what I'm showing you here is sort of some time-lapse microscopy of the expression of one of these folding mutants, these Brachos folding mutants. And you can see that they starts out with sort of a, beginning to form some cellular aggregates, and it just forms more and more and more and more aggregates. And eventually this is apoptosis. You can see sort of the blebbing here, having it sort of literally fall off the dish. They're insoluble in detergent. So they really are incredibly aggregation prone. What I've shown you up here is, again, the formation of these aggregates is associated with many aberrant cellular responses. And this is an Exon 5 staining. This is caspase 3 on a Western blot. So again, these will induce apoptosis if expressed for a while. Along the way, and we'll talk a little bit more about this because this is where the signaling, I think, is in terms of trying to preserve function. You can activate ER stress, and a variety of endpoints have been used to show this. And down here, I pulled this out last night. I'm a little embarrassed given all the quality of the imaging I just saw this morning, but this was an attempt to show that these actually will induce the expression of these mutant proteins will actually impair the UPS, or the ubiquitin proteasome system. So if you take GFPU1 cells, which is basically GFP with a degradon attached to it, so that it normally, under normal conditions, is continuously degraded by the proteasome. And if you add lactocysteine, you can see all the cells light up. If you co-transfect in a mutant SPC, but not the wild type SPC, you can see now they start the expression of green fluorescent protein, and the cells are actually transiently transfected with the mutant protein. So this has the capacity then to to actually inhibit the proteasome. And this is important for disease because a lot of the environmental exposures that we see, cigarette smoke, viral infection, also affect the proteasome. So you can imagine, a patient with one of these mutations and then who also smokes, who also has environmental toxins or exposed to ozone, and really can really sort of trigger some of this dysfunction that I showed you here. So let's see. Okay. So how does self deal with this? So classic, as you might think, is the activation of the unfolded protein response. And so what is that? So as many of you in this room are aware of, there's multiple sensors within the lumen of the ER. The three major ones are IRE1, PERK, and ATF6, and they're all held silenced by the binding of BIP to their luminal domains. But in the presence of large amounts of unfolded protein, they basically compete for BIP binding and then can activate and activate these. They're activated by different mechanisms. IRE1 dimerizes after being phosphorylated, and then oligomerizes, and then has two functions. One function is to actually promote an RNase activity to facilitate XBP1 splicing. And XBP1 splicing is incredibly important for a number of adaptation genes, mainly to expand ER folding capacity and chaperone transcription and translation. The other side of this, probably because we're multicellular organisms, is not a favorable one for the organism, but through its kinase activity engages TRAF2 and can, through a large engagement of multiple different MAP kinases, JUNC, SARC, I'll show you in a second, can actually send out alarm genes. So you can actually promote under duress the release of cytokines and chemokines, again, telling you that I'm a distressed cell. So there's sort of a teleological reason for this to happen. It's also been shown to activate NF-kappa B. PERK actually does the opposite. Instead of increasing folding capacity, it tries to attenuate translation, predominantly by phosphorylating eIF2 alpha, and then selectively activating transcription factors such as ATF4, again, for adaptation. So turning off protein synthesis and helping with the adaptation. And ATF6 is really not well known. There's not a lot of great reagents for this. There's not a lot of great antibodies, but it seems to be protective. It seems to work in concert, and I'll show you that a little bit with IRE1 and XBP1 to help, again, with the adaptation. And what we've shown is that in a variety of studies over the years, is that the expression of these proteins, both in transient transfections, as well as I'll show you in a second in vivo, can activate all three arms of this unfolded protein response and can, as I say, trigger apoptosis as well. So here's PERK phosphorylation of the eIF2 alpha shown down here. And I would note that most of this you'll see with the folding mutants and not with the trafficking mutants. So it is fairly specific. We use that sometimes as a control for this. And then, of course, you have IRE1 here. And these are the alarm genes. We've got NF-kappa B activation. We've shown that JUNC is up, and we show that we can selectively activate multiple cytokines. I just picked IL-8 out of here just from an old data set. So all of these are in play in terms of the adaptation to this, in terms of the epithelial dysfunction. And does this matter in vivo? So it turns out that if you look into biopsies of patients either with the familial forms of SPC-induced interstitial lung disease or the sporadic forms, you can find evidence for a lot of these pathways. This is protein aggregation in amyloid. This is Congo red staining in the lung of a patient with an SPC Bricos mutation. These is a familial case published from the Vanderbilt group showing activation of ER stress staining in the epithelial lining cells for BIP, as well as some of the key chaperones. This was XBP1 mediated chaperone EDOM, and again, staining for actually XBP1 protein shown here. And then this is a study out of Germany that actually isolated type 2 cells from patients with IPF or COPD and other disease controls. And again, selective activation by the IPF epithelium of XBP1 splicing, PERK here, not shown as ATF6, which was also activated in this. And of course, the induction of apoptosis, CHOP, and the loss of procaspase 3 shown down here. So all of this together. So the biology over the years taught us that these mutations might affect the fibrotic response by at least acting through aberrancy of the UPR and disruption of the ubiquitin protease subsystem. And so how to prove that? So we did what everybody did. We came back and convinced the university to give me some money to make a mouse. And so we made 2 mice. I'm going to show you them in sequence. So we decided to look at the folding mutants, and we had to do a little bit of a trick here. And I'm glad that we did this because we did this about 4 years ago when it was mostly ESL technology based, where you had a targeting vector that you knocked it into ESL, screened them on the basis of a neomycin cassette, which in this case we had put into one of the introns, and then flanked this with LOX-P sites so that we could then cross this to a deleter mouse. What I won't show you is the trials and tribulations of the postdoc that initially did this with a constitutively active crate, for which we got no live mice. So the expression of these proteins, the patients, I should say, are all heterozygotes. But even the heterozygotes, we have no live mice. And so it really portends to the toxicity of this, even in a developing lung. So our next step was to actually basically just cross this to a ROSA 26 ERT2 crate. And again, this is only expressed in type 2 cells, so we don't have to worry about tissue specificity here, and we could take advantage of that particular crate. And so when you add tamoxifen to adult mice, and within 7 days, we get transcription and translation. This is the primary translation product of the SPC. In this case, we picked a clinical mutant, C121G, which was obtained from a patient in Texas, for which we also have the iPS cells now, which we can talk about afterwards. But again, here, what you'll see is that we get primary translation product and no processed bands in the type 2 cell, whereas the wild type protein is processed. We get no mature SPC in the mutant, and again, suggesting that this is not trafficked beyond the ER or the Golgi. We confirm that by showing ER retention and staining with the marker, in this case, KDEL. And we activated all of the unfolded protein response. We got activation of BIP, we got activation of ATF4, we got XBP1 splicing shown down here in a PCR-based assay, and we got evidence for CHOP activation as well. And what's most important is we were able to mimic the fibrotic phenotype for this. So this is a disease phenotype. This is a fibroblastic focus. I say that with confidence because we have staining for both SMA and surfactant protein B in red. And you can see the juxtaposition of myofibroblasts adjacent to epithelial cells. This is Pipocerius red staining. This stains for fibrillar collagen. We have the presence of fibrillar collagen, even in areas that are not as fibrotic as this. So we're starting to see fibrillar collagen deposited in the septa of alveoli, even before they collapse and coalesce into this kind of foci here. You can measure soluble collagen, you can measure hydroxyproline. I'm showing you some of this, and we get evidence for physiological restriction, which is what the patients have, which is the downtrending of the pressure volume curve, and this is the lung compliance measurements shown here. So this is proof of principle that these mutations expressed exclusively in the type 2 cell can drive a fibrotic lesion downstream and produce a disease model. We've refined this a little bit. I won't spend a lot of time on this, but there's a lot of mortality when you do single dose IP injection of tamoxifen. So we've actually, Jeremy Katzen has now taken this model and actually stretched it out with weekly oral gavage treatments of this. And so what's very nice about this is we have very little early mortality now. And what you'll see is that by about 4 weeks, they really start to fall off the curve here in terms of their weight loss. And if you look at 8 weeks or 7 weeks, we get the same sort of defects. We get fibrillar collagen, we get evidence for infiltration with inflammatory cells and myofibroblasts. This is the quantitation of the PCR. We have gene expression for collagen genes, 1A1 and 3A1 shown here. And again, restrictive lung physiology. And importantly, he gets all of the signals of continued ER stress. And so that's shown here. He's got PCR here for VIP expression, XBP1 splicing, ATF4, and CHOP expression. So the model can be done in a persistence model and used to sort of interrogate this pathway both in an acute setting and in a chronic setting. So I want to talk for just a second about proteotoxic stress and what could be going on here. So we think that obviously that we've created a dysfunctional cell, but how is this happening? And is it one cell type or not? And so here's an example just from the in vivo models, again, where we have evidence for apoptosis. Years ago, we did a study with Aberrant where we expressed some of our surfactant mutants along with multiple different doses of tunicamycin and other ER stressors, other chemical ER stressors. And under low dose ER stress, instead of getting apoptosis, what Zia and her colleagues found was actually that we got the activation of a program for upregulation of the mesenchymal component expression, downregulation of things like cadherins for epithelial markers. So for all the world, the cancer world, this is EMT. We're not allowed to say that in lung biology anymore. And she was actually able to show that this was at least mediated in part through a Src kinase. And so, as many of you may know, Src kinases are now in clinical trials for both cancer, and there's been some equipoise to do this in IPF based upon a recent paper from Greg Downey's group in the States. And so, so we have at least a couple phenotypes. We have an apoptotic phenotype, we have an EMT phenotype. And really what our working model is, it goes even a little bit deeper than that. And that is these different defects in quality control. I've showed you mostly activation of the UPR leading to stress. There's also a mitochondrial phenotype of this, which we won't talk about today. But there's probably multiple endophenotypes within the lung. And we mentioned senescence, we mentioned apoptosis. We recruit inflammatory cells through elaboration of things like CCL2. And again, these things will make things like CTGF. And so there's this pro-fibrotic phenotype. And what I want to spend a little bit of time on today is also what happens with it. And I know that people here are very interested in AT2 to AT1 differentiation. Well, there's a little intermediate along the way called the transitional cell, which seems to be exaggerated again by the expression of some of these mutant proteins or the adoption of ER stress or metabolic reprogramming. And so the heterogeneity in this was initially fueled by seminal studies that were done in patients on the left with IPF. This is work from John Kropsky's group at Vanderbilt, which looked at single-cell RNA-seq and was able to identify among the epithelial components, not only type 1 cells and type 2 cells, but what the arrow I've drawn here in this very faint color here is the presence of a transitional cell state, which was KRT17 positive, KRT5 negative, and had a high expression of various markers for profibrotic, things like V beta 6 activation of TGF beta. The mouse equivalent of this, shown over here, was initially described by Rachel Siemens, but has been also put forth by Herbert Schiller in Gießen, that in a bleomycin model of lung fibrosis, you get the emergence of KRT8, Claudin-4 positive cells as well, and a failure to differentiate into type 1 cells. Rachel showed this in influenza as well as in a bleomycin model. So various injury models will induce this transitional cell state, and we know that even after viral infections like COVID-19, there's an incidence that's not too small of patients that actually undergo fibrotic remodeling. And so we started to look at this, from a granular standpoint. And so what we did was first isolate type 2 cells from the model at an early time point. In this case, 5 days, 14 days, and actually 28 days, it's a typo there, and did bulk RNA-seq. I'm going to show you data from the 14-day time point, because this is the time point that actually is the transition from inflammation to fibrosis. This is when you start to see fibrillar collagen. And actually, if you look at both genotypes, that is the folding one, which we've talked about, as well as the trafficking one, there's a huge overlap in commonality of gene expression. Over 3,000 genes are upregulated within 3 days and stay up through the entire period. Huge amounts of changes in pathways related, again, to reprogramming to some sort of a mesenchymal phenotype. I've shown you here some of the genes for both of the mouse models, and you'll see that there's also evidence of senescence with CTGF, SASPs, and SERPIN1, as well as B beta 6 and this integrin beta 5 shown here, et cetera. So there's a reprogramming here. Coupled with this, as you might expect, is a loss of the alveolar type 2 program. And this is work that Jeremy Katzen has recently published in PNAS. He took the same bulk RNA-seq, interrogated it for the type 2 cell program, and noticed to a protein or to a gene, downregulation of almost all the genes associated with normal surfactant function. All the surfactant proteins, the enzymes that process them, the aspartic proteases, confirmed this, validated this with Western blots for some of these, like surfactant protein B. Again, now down as early as even 7 days, shown here. And at the same time, we started to find upregulation of some of these cytokeratins and the things that were associated with the previously mentioned transition state. And so we had this hypothesis that could this be the UPR that was actually driving this phenotype towards a transitional cell state and the adoption of this intermediate shown here. And so to do that, then we did the big experiment that everybody does, is that we then did single cell RNA-seq. We did this at 3 days, we did this at 7 days, we did this at 14 days. I'm showing you the early data because it's the most interesting. And in terms of identifying the clusters and then focusing on the epithelium, only showing you the epithelium. This was an EpCAM enriched population that was CD45, CD31 depleted. And then out of this, focused on the distal lung cells, which were the type 1 cells and the type 2 cells. And as early as 3 days, he could find evidence of activation of a UPR program in some of these type 2 cells. So you have homeostatic type 2 cells. They're the ones that actually express, sort of the markers of surfactant. You had the UPR, which I'm showing you here, so for one of the heat shock proteins, as well as CHOP, shown here, upregulated as early as 3 days, and the adoption of a transitional cell state. This persisted through day 7. He then did some RNA trajectory analysis using pseudotime. And under normal conditions, when you combine the clusters from both the wild type and the mutants, you actually see that you can go from a homeostatic type 2 cell through this to a type 1 cell. But there's a block. We can't do the lineage tracing yet because we're in the SPC locus. So we've actually developed something related to the ABC locus now that we can knock in a cree to actually do the reporter lineage trace. But we did the old-fashioned thing, which was staining for the markers of SPC and KRT8 and a lot of cell counting. And what he was able to show is that you start out with mostly type 2 cells expressing only SPC. And as you progress through this, you end up with mostly cells that are sort of mixture of either double positive or KRT8 positive. So this suggests that, again, we're impairing the ability of these cells to act as stem cells for repair. And so then the question was for him, could he prove this? And so we had to come back with some drug testing. We don't have high throughput screens here. There's a lot of finding, Ryan, I have to say, Lucas. So we took a candidate approach to this. We hypothesized that this was probably based upon some data from the gut, that it was IRE1 and XBP that could be mediating this. And so we went back and we created a cell line that was stably transfected with and inducibly expressing one of the mutants that was in the mouse model, C121G, and you can see here that addition of doxycycline will turn that on. And then we interrogated all 3 arms again of the UPR, but we mostly focused, well, first we wanted to show that it did what we said it was going to do, and it does in fact induce ER stress. It does activate XBP1 splicing. It does affect XBP target genes such as EDM and ERDJ4. It does activate ATF6 activity, and this is a reporter construct shown here, a luciferase reporter construct, and it does actually result in the phosphorylation of EIF2 alpha. And so all 3 arms are activated, so we could interrogate it with anything we want. We've put PERK inhibitors in here. We can knock down PERK. We've done things I'll show you in a second with ATF6. But what he focused on here was we had a collaboration with a company that was formed off of Feroz Papa's observation about Kira compounds, and the Kira compounds were developed initially for an overstressed pancreas, diabetes by NUCSF by Dr. Papa, and the company came out of this. There were tool compounds. So Jeremy went and tested the lead compound, which was OPK711 for the company, the lead tool compound, I should say. And this is all published now, so I'm not telling you anything confidential. And so you can see that in our hands, we get XBP1 splicing with the mutant, and we got a dose-dependent decrease here with the use of OPK711. I mentioned Orin 1001 because of those of you in the cancer biology arena know that this is also another IRE1 inhibitor. It acts specifically on the RNA, so it blocks XBP1 splicing, but does not block the kinase activity. Cures work by actually affecting the dimerization and the phosphorylation, the kinase activity, which then sort of prevents the RNase activity sort of secondhand. But you can see that Orin works just as well, but as I'll show you in a second, did nothing in vivo in terms of the phenotype. And so we think that this is the kinase activity is what's important for this. And so what Jeremy did first was he went into an organoid model. So this is isolating alveolar cells out of the SPC mutant mice, not induced, putting them in organoids in regular medium, and then adding 4-hydroxytamoxifen after they had formed their organoids, and then in the presence or absence of the IRE1 inhibitors. And very nicely, you can see that we get upregulation with tamoxifen, at least 10-fold, 20-fold increase in SPC expression. We get activation of XBP1 splicing shown here, and we get activation of markers of the transition state, KRT8. And with all of those, except for SPC, IRE1 inhibitor OPK711 works beautifully. It knocks down XBP1 and KRT8 and doesn't affect SPC gene expression. So armed with the MLE12 data, the organoid data, we went back into the mouse model and an acute model to see at 7 days, could we hit target and could we affect some of the function? And in fact, what you see here is we dosed it simultaneously with the induction of the mice by a single dose of tamoxifen. And at the end of 7 days, we were able to rescue the decrease in SPB, the AT2 phenotype. We were able to knock down XBP1 splicing. So yes, we hit target. And we were able to reverse Claudin-4 and KRT8 expression and mitigate some of the lung injury that you see at 7 days. So I think, taken all told, this would suggest that that this is a viable pathway to interrogate to try to mitigate both either the early lung injury and possibly the fibrosis. We've tried this in the chronic model. This is a tool compound for a reason. It actually does cause cardiomegaly. So there's something about IRE1 inhibition and cardiac function that still needs to be worked out, at least in terms of the compounds that are available to us right now. And so where do we go for this for future directions? We're very interested, again, in trying to modulate the UPR. We think that knocking down IRE1, the reviewers at PNAS made us say that it was XBP1, and we had some bioinformatic data. But based upon the data I didn't show you with the Orin compound, I think the kinase activity is actually almost as critical, because that's the one that will engage SARC and JUNK and some of the other ones that could do some of the reprogramming. And so, so where we go now is we're actually looking at whether we can expand proteostasis. So we've gone back to the MLE12 model. And I put this slide in because we were talking about ubiquitin-mediated degradation yesterday, and whether or not modulating ubiquitin activity and degradation of the mutant protein was a viable option. The problem is that you need to have the degradation of these proteins. If they sit in the cell long enough, they aggregate, and that's shown here. We actually added iristatin, which is an inhibitor of VP97, so the retrotranslocation that takes mutant proteins out of the ER. And you can see what you go from an ER expression pattern shown here to just basically aggregates. We think these are intra-ER. We have to get some better imaging to prove that. And so that would suggest that we've got toxic aggregates sitting in the ER. And you can see biochemically that we induce the toxic aggregate shown here. So modulating the ubiquitin proteasome system may be a little bit tricky here. And we're actually turning towards thinking about ATF6. ATF6 is interesting. It's requires its processing in the Golgi once it's disengaged from the ER. And we actually, as I mentioned, we actually have a large amount of ATF6 activity that's inducible. We can knock this down with inhibitors of ATF6. And there is an ATF6 agonist, which is equally efficacious as the Sftpc mutant itself in inducing ATF6. And a very preliminary unpublished data, we went back to the EEY iristatin treated aggregation model. And what you can see again, under control conditions, without iristatin, we just have single protein product of mutant Sftpc. You add the iristatin, and this is a cyclohexamide chase. This replaced all the S35 I did in the lab. And you can see that under control conditions, there is actually some degradation of the protein over time, but that in the presence of iristatin, we do get intracellular aggregates. Adding an ATF6 agonist actually decreases the aggregation and enhances the degradation of this within the ER. And inhibition of ATF6 actually promotes aggregation and delays that, and the quantitation of that is shown down here. So targeting ATF6 with some emerging drugs and tool compounds, I think, is an option as well in terms of trying to fix the proteostatic defect. We also have a program looking at PERK and EIF2 alpha, which I won't have time to share everything. It's really Jeremy's story, so I'm gonna leave that. If I have a couple more minutes, I can just go through a couple more things that are sort of proof of concept. So this is the other mutation, the trafficking mutation. This is I73T. And this was worked by 3 different postdocs. So it was a long, arduous project. So the answer to the trainees is don't give up. It'll eventually get through the system. Pretty much the same idea, except that in this case, we had flipocytes. And so we actually had the neomycin cassette to produce the hypermorph in exon 1 shown here. Again, add tamoxifen, rapid increase in mRNA, some early mortality, which is dose dependent. At 2 weeks, you have an inflammatory phenotype. These are the little mouse CAT scans shown here, showing the patchiness of the disease. And then you get eventually fibrosis at 4 to 6 weeks down here, collagen gene expression and all of that. So why am I showing you this? So I showed you a whole story about IRE1 reprogramming of type 2 cells. Well, it actually happens here. Remember, these don't induce a lot of ER stress. So there's got to be more than one cue to reprogram the cells. And this is single cell RNA-seq that we just unearthed about 2 weeks ago. And this was from a study where we actually CD45 depleted them and then added back 20% cells. We had a more balanced approach to this. But I'm going to show you just the epithelial cell data shown here. But because when you actually cluster these again for epithelium, you get exactly the same sort of interesting thing. That is, you get the homeostatic type 2 cell shown down here. You get a type 1 cell shown over here. And we actually found 2 transition states shown here, an early and a late transition state. And you can follow the pseudotime using these sort of gene markers and show that you go through this, KRT8 state. But what's interesting is if you actually take and disengage the clustering for all the time points for the model from 0, 2, and 4 weeks to just individual time points, you can see that uninduced control mice have type 2 cells and type 1 cells. And as you go through this, you start to get this accumulation of all of these intermediates. You lose the type 2 cells, but you don't pick up the type 1 cells. Now, not quantitative per se, but very intriguing. And Willie Roque Barbosa, who's actually taken this project on, has done some of the quantitation for the relative amounts of this, and you can follow along with the colors. But it would suggest, again, that we have the same types of transition cell states for this, but the mediation of this might be different. I'm going to skip through this only to tell you that there's a metabolic phenotype. And the metabolic phenotype involves a Warburg effect and a loss of mitochondrial function. And we come to the end. And so what I hope I can convince you is that we've been able to use very rare variants in surfactant protein C as tools, as substrates to sort of probe cell quality control. In the case of what I mostly have showed you today, it's been the proteostasis network, the UPR, and the ubiquitin proteasome system. The second one actually has a macroautophagy defect, and so the signaling for that we think is more metabolic. But regardless of this, these have been useful tools to actually probe the various appearance of these various phenotypes. And again, we can find these signals in the lungs of IPF patients. Whether all IPF patients have them or whether there's endophenotypes remains for the clinicians that get samples and carefully do this. But I also want to make you aware that this is not the only models. And so there are other models also associated with quality control. So Zita Boric published a GRP78 loss of function model. It has a model of fibrosis. There is the recurrent low-dose bleomycin model, which is a little different. It's less injury and more fibrosis and a little more sustained, shown here. Defects in telomere, short telomere syndrome. So this was work done out of UCSF and Paul Walters with a TERF1 model, one of the telomere maintenance genes. Again, the appearance of fibrosis, not in this time scale that we saw. This is now months shown here, but again, potentially useful. And then this is the senescence model for those of us who are into the senescence. I think this model is freely available. Barry Stripp's a pretty giving guy. And so the SYN3A, which is a repressor of cell cycle, so the cells are stuck in G1, end up with progressive fibrosis over about 6 to 8 weeks and have some of the same features as our models in terms of elaboration of TGF beta, and Barry's actively testing compounds right now in this model. So these would also be sort of available as well in terms of thinking about how you would want to model some of what you're looking at. And so with that, I have to thank all the people that do the work. Talented postdocs, Luis Rodriguez and Willie Roque Barbarossa and Aditi Murthy are the 3 postdocs in the lab right now. One is our immunologist, one is our fibroblast guy, and one is all things, Luis. And we couldn't have gotten this done without him. A lot of collaborators worldwide. There are people in Europe that have sent me samples and sequences for SPC mutations. And we have a great lung biology institute here at Penn with Ed Morrissey. So I'll stop there and take any questions, and thank you for your attention.